And we'll do our final system, which is going to be the reproductive system. Again, we always start with what are the main roles that the system plays. So, in the terms of reproductive system, what is it doing for you?
Produce offsprings.
Yeah, so it's producing offspring. Um, Although there's easier ways to do it than the way we do it. There's a lot of animals that just like make little clones of themselves. If you were a sea anemone or a hydra or something, you could just start growing off a little part of you and bud it off, and then it could become its own free living organism. And there's no waste. Most of the sperm and the eggs that we make never become new offspring. They're wasted. A lot of wasted energy there. There's the big complicated social dance that you got to go through trying to get the sperm and the eggs actually connected up and all that. Um, what do we gain by doing this kind of sexual reproduction that we do?
Genetic variation.
Yes, we have genetic variation, and it really is worth just spending a few moments just kind of emphasizing how much genetic variation makes a difference. It's not just about like obvious physical things like some people are taller or shorter or have this color hair or that color hair or bigger nose or smaller ears or whatever. Um, there's all sorts of um, alleles that change the way our metabolic processes work. Right? Some people are lactose intolerant. Some people eat ice cream and cheese just fine. Some people drink alcohol and they get all red and flushed and it's a really bad thing. Other people, it's not such a bad thing. Um, Even in terms of personality types, people who study addiction and stuff have found like there are genetic markers for like impulsivity. When they do these things of these tests like how much can you put off something for now for a greater reward a little later down the road. There's a genetic marker for whether somebody can just kind of sit and like wait for the bigger reward down the road versus nah, I just got to get this thing right now. um, they found a genetic marker for just how likely you are to just take advice. Like they had kids like taking some exam, and people would give them advice, and sometimes the advice was either helpful, sometimes the advice was actually leading them astray. And what they found was there was a genetic marker for some people would listen to the advice, either do better or worse depending on what kind of advice they were getting, and that would influence their behavior and their performance. And other people, it didn't matter whether or not somebody was offering them good advice or bad advice. They kind of just did whatever they were going to do anyway. So just kind of giving you this sense of you know there's a huge array of um, metabolic behaviors, physical characteristics, even personality things that are all connected to this genetic variation, which is pretty cool. You know, so let's get down into kind of how we do it. So when we talk about the primary sex organs, what we really are talking about are just the gonads. The ovaries and the testes. Um, 'Cause these are the places where you're actually making the gametes. Um, what is the special kind of cell division that happens in the gonads?
Meiosis.
Meiosis. This is where we're going to find meiosis. This is the only place in the body where you find meiosis going on, is going to be in the gonads. We'll talk about meiosis in a more detail in a few moments. Everything else are kind of like the accessory organs. Accessory organs are basically going to be the things that, allow you to hook up the gametes, the penis and the vagina and all that. And places, the thing that the female reproductive system does in addition to making gametes and connecting them up is also providing a place to nurture the development of the fetus. So, we're going to talk about the uterus and all of that. But when we think about at the core of things, it's going to be ovaries and testes. And that's kind of where we're going to start with looking at how, first, kind of what are gametes? How are they made? How is their production kind of regulated by the body? And then we're going to get into all the other stuff as well, obviously. So let's talk about meiosis. What is meiosis? Mitosis is kind of more the classic cell division. How is mitosis and meiosis different from each other? They both start, there's some cell that's going to start doing some cell division.
I think mitosis does the identical split.
It's gonna go, if you remember, every chromosome is going to duplicate into two sister chromatids, and then every single one of those sister chromatids lines up along the metaphase plate and gets pulled off to both sides. So you end up with two identical daughter cells. And yeah, remember, there are 23 maternal chromosomes and 23 paternal chromosomes. What did I write? So 46 in total. So we call this is 2n. These are also 2n because there's 23 chromosomes that make up kind of the human blueprint. There were 23 that you got from your mom, 23 that you got from your dad, different versions. Those are those alleles, right? Your genotype. Your genotype is which allele of that chromosome or of that gene do you have from your mom, which allele do you have from your dad. So we start with a 2N cell. We end up with two, again, this is called diploid. So we end up with two identical daughter cells, and they're both, diploid, just like our cell that we started with. And this is basically all growth and repair. So almost all of the cell division going on in your body is just your kind of regular old mitosis. So meiosis, this is only in the gonads. And how is it radically different? It's going to start with a 2N cell here. How is it radically different from mitosis?
It will end with two haploid 1N cells.
So it's going to end up with haploid daughter cells. How many haploid daughter cells?
Isn't it 4? Four?
Right, this is going to go through two cell divisions. This is going to go into the first cell division and then into a second. There's meiosis 1 and meiosis 2. So we're going to end up with 4 haploid daughter cells. And so that's different. Instead of 2 diploid cells, we have 4 haploid cells. How else are these daughter cells different?
Genetically different variation.
Yeah, so these are genetically unique. Right. In that process, again, I don't have the time to go into all the detail, and hopefully you remember it a bit from your Bio 110. But there is the independent assortment during metaphase 1, where you're pulling either mom 1 or dad 1 and mom 1 or dad 2. You basically, for every chromosome, there's a maternal and paternal, but either one or the other is pulled to one or side or the other. You don't pull copies of both like you do in mitosis. So you have this independent assortment where you're pulling only the maternal or the paternal version of the chromosome to one side or the other, as well as the fact that during prophase 1, you've also got the maternal and paternal chromosomes lying on top of each other and swapping out parts of each other and crossing over. So you've got like these two major things happening that are basically kind of shuffling the deck between the maternal and paternal versions of your chromosomes. So, here you have, again, I said maternal and paternal versions of the blueprint. Each of these daughter cells is some weird mix, unique mix of both of those that you are then going to contribute to be your maternal or paternal version of the blueprint to your offspring. So these are going to be the sperm or the eggs. And again, if you have a haploid daughter cell, these are what we call 1N. And then when I have a 1N sperm coming into a 1N egg, when they meet, that's going to be a new 2N, the zygote. That is going to be the beginning of the new little being. Right? So there's going to basically be meiosis to create a 1N sperm and a 1N egg. And once those two come together to become a 2N zygote, that is now going to just undergo mitosis, to grow into an embryo, into a fetus, into a baby, into a toddler, into a teenager, into an adult, whatever. All right. So let's spend a little bit more time looking at the actual zygotes. I mean, not the gametes. So I'll introduce them, and then we'll kind of start going down kind of two rabbit holes, one for the male and one for the female. Obviously, for the guy, you've got the sperm. Sperm are really dinky. They're basically kind of a delivery system for genetic material. There's kind of a nucleus with the DNA. There's this little mid piece here with lots of mitochondria. Um, and there's the flagellum. This is the only cell in an adult human that has a flagellum. And the flagellum is basically like a big oversized cilium. It can wiggle back and forth, and its job is basically to propel the cell through, through space. So it basically lets this thing swim. Mitochondria to make the ATP to power that swimming. We're going to see when we look at semen later on that part of semen is sugar, right? So these guys got energy to do their swimming. We're going to see sugar getting produced by the like seminal vesicles, during the whole ejaculation thing that we'll look at later. The egg, on the other hand, is huge. So it's one of the largest cells in the human body, supposedly visible with the naked eye even. And it's got to be huge because once the egg is ovulated, and it gets fertilized, it spends almost a week drifting through the fallopian tubes before it ever gets down to the uterus and implanting in the wall of the uterus where it can now start getting nutrients from the mom. So the egg has to have enough kind of resources to survive the first week of embryonic development completely self-contained. So we'll talk a little more about that later. But basically, it's got lots of nutrients, and it's also got all the organelles that the new baby is going to ultimately, the cells of the new offspring are going to have. Right? The sperm is basically just a way to deliver genetic material. There is not much more. In fact, we'll talk about during spermatogenesis, almost all the cytoplasm is spit out, and it's basically just like it's like a nucleus with a little envelope with a propeller on the back. All the mitochondria that are in your cells and in every one of your cells, they're all matrilineal. They're all from your mom. They're not from your dad at all. I mentioned last class that the ovum is never found alone. I talked about the idea of the follicle, that the ovum is always surrounded by these other cells. They're follicular cells, granulosa cells. We'll talk about that in more detail. Even after the egg is ovulated, it still has these cells surrounding it, which we're going to call the corona radiata. So these are these cells that are surrounding the egg here. And what that means is when the sperm finally, it's like, all right, made it to the egg. It's like, oh no, just hit a brick wall here. So the sperm, the one other thing the sperm has in addition to kind of like the propeller and the mitochondria, it has enzymes that help it break through that corona radiata, that wall of cells, so it can actually reach the egg. And that's going to be called the acrosome. So they're kind of in the head. Kind of just so once this dude gets there, it starts breaking through here. Um, I'm not going to go into the detail, but there's actually really cool stuff. Once a sperm actually reaches and gets to the actual egg, and the nucleus actually starts fusing with it, then it triggers these, they call them blocks to polyspermy. As soon as this happens, there's this kind of gelatinous layer that starts hydrating, and basically every other sperm that was coming in trying to be the one basically gets pushed back and detached off. So only one sperm. Once one sperm actually fertilizes it, it starts this whole process that basically pushes away and keeps all the other sperm from getting there. So that's the basic idea of the gametes. What we're going to do now is look much more detail at how are sperm actually produced? How is their production regulated? Then we'll look at the egg, and we'll look at the development of the follicle and what's influencing that. And again, when we talk about the sperm, it's going to be simpler. They're just kind of getting made and kind of ejaculated. Whereas the egg is going to be a much more complex story because it's going to be in conjunction with the uterus. It's not just about making a gamete. It's also about preparing the possible development of the fetus, the embryo and the fetus. So, all right. So let's. Oh, I keep hitting the wrong thing here. All right. Let's start with the male. So first I'm going to describe spermatogenesis briefly, and then we'll kind of go in down into the testes and look in more detail what's going on. Spermatogenesis is just the process of making sperm. You start with this cell called a spermatogonium. These are just sitting in the little tubules in the testes. We'll look at this in a little more detail. So these are just always there. And spermatogenesis starts with mitosis, where this thing splits. And it has two daughter cells. One daughter cell is just replacing the spermatogonium, so we're ready to go for another round, right? Because this just goes on for a lifetime. Although it does slow down. And then this other one gets committed. This is going to be kind of my primary spermatocyte, which is now commits into meiosis. And then this is going to do meiosis. This is going to do those two. You have secondary spermatocytes. You have these things called spermatids. So these are and again, if we were in anatomy, you'd have to learn the names of all these. I don't care if you know secondary spermatocyte and spermatid. If you want, you can learn it. But basically, you end up with these 4 haploid genetically unique daughter cells. And then there is this kind of radical remodeling that happens. Again, you lose most of the cytoplasm and you grow a tail. This last step, this remodeling, they call spermiogenesis. In a little while, we're going to talk about the blood-testis barrier. One of the reasons you have this kind of barrier between the insides of the little tubules in the testes and your blood is these cells are so radically different from one of your normal body cells. You need to kind of hide them from the immune system because they've become very kind of unique compared to a normal cell. So this is this process. And again, it's kind of constantly happening. This spermatogonia undergoing meiosis, replacing itself. Then this one here is ready to go for the next round. This can do the same thing again and again. This is going to be very different when we look at oogenesis in the woman. In the woman, all of her oogonium have already committed into oogenesis before she's born. So by the time she's born, she's just got primary oocytes that are already stalled out in meiosis. So like every egg that she ultimately ovulates had already committed and started oogenesis in meiosis when she was born. So again, which is different. In the guy, there's this just kind of constant production. So again, we'll get to oogenesis in a little bit. All right, so all of this is happening in the testes. So let's shift our focus into the testes. They get their name from the root to bear witness, like testify, right? You go testify. Like back in the old days, instead of putting your hand on a book when you're going to swear in front of a jury or something, you put your hand on the family jewels. So testes are to bear witness, to testify. Most people know what a testicle looks like. They're sitting in the little scrotal sac. Usually if you put little pubic hairs on there, people know what you're talking about. Why are the testes sitting outside of the body? Something so critical to the survival of the species. Why is it hanging in such a vulnerable place?
Isn't it something about temperature?
Yeah, exactly. The spermatogenesis that we just described only functions well if that's a few degrees below body temperature. So the testes are kind of hanging out and getting kind of air cooled outside of the body. We'll talk a little more about this when we put the testes into the larger context, but you basically have muscles that can pull them up closer to the body to keep them warmer, drop them down lower to cool them off more. Other muscles that can wrinkle the skin to provide more or less surface area to radiate out heat or conserve heat. So your body's kind of adjusting the temperature of them. The testes have two main things going on inside. One is sperm production. The sperm production is happening in these little tubules. You've got all these little tubules called the seminiferous tubules. And they are where sperm production is happening. They will ultimately all dump out into this kind of common area, the rete testes. Then they go into another tubule that's kind of sitting on top of the testes, although still in the scrotal sac, called the epididymis. This is kind of sperm kind of stored and mature here. This is like it's, and then ultimately, obviously, if you're gonna get the sperm is part of the semen, part of the ejaculate, you've actually got to get them up into the body cavity where they can, enter into the urethra, so they can come out the tip of the penis. So then we have a big tube that's going to go up through a little tunnel through the inguinal canal and enter into the abdominal pelvic cavity. So this is going to be the vas deferens. Also called the ductus deferens. This is to abdominal pelvic cavity. Actually, I can't spell. Right, so ultimately the sperm are going to join up with all of the other secretions to become semen. We will get there in a little bit. I want to kind of stay in the testes for right now. So one thing, a major thing happening in the testes is sperm production. Sperm production, which is happening within these seminiferous tubules. Again, we'll look at that in detail in a few moments. The other important thing going on in the testes is testosterone production. That is happening in these little cells that are located in the loose connective tissue in between the seminiferous tubules. These are called the interstitial cells of Leydig. Interstitial, again, just meaning in the spaces in between. So these are cells kind of in between the tubules. These make testosterone. We should talk a little bit about the control here. What hormone stimulates testosterone production?
Luteinizing hormone?
It was luteinizing hormone. So what I'm gonna erase this here so I can have a little more place to draw. So if we want to kind of understand the kind of control of this, we're going to have to think about the brain. So let's put the brain in here. The hypothalamus and the pituitary gland is not in your pituitary gland's kind of like right here. So you're going to have gonadotropin releasing hormone up in the hypothalamus, which is going to talk to the pituitary gland, which is going to release luteinizing hormone, which is then going to talk to these guys. So the basic idea here is in a guy, so we're talking about a guy. In a guy, gonadotropin releasing hormone is released continuously. It pulses out every few hours. This is going to be super different than what goes on in the woman. In the woman, gonadotropin releasing hormone is going to rise and fall in a monthly cycle. In a guy, it's just every few hours it pulses out. That gonadotropin releasing hormone, whoops, gonadotropin releasing hormone is then going to stimulate the release of luteinizing hormone. Luteinizing hormone is then going to stimulate the production of testosterone. And then we're going to have a feedback loop that happens here. If there's enough testosterone, the testosterone feeds back. I'm putting a big minus sign here, is it suppresses the release of the luteinizing hormone. So if there's enough testosterone, the release of luteinizing hormone will be suppressed, which means you're not going to be stimulating the interstitial cells to be making so much testosterone because we got enough. So this is an example of one of these negative feedback loops to regulate the levels of the hormone. We'll see something a little different happening when we get to the sperm. But so, does this make sense? I should mention also.
I do have a question. So it makes a ton of testosterone, and then the negative feedback loop slows all the production down. So then ultimately you get lower amounts of testosterone.
No, you just get a kind of a homeostatic level, right? If it drops
Okay.
down, then you release the inhibition, so then we start making more again.
Gotcha.
Make enough, then it it's like if you're driving, you're trying to stay like 65 miles an hour down the highway, and you're slowing down. You hit the gas until you hit 65, then you take your foot off the gas, right? But if you're dropping below, you're going to hit the gas. But if you're getting too fast, you'll take your foot off the gas.
Mm-hmm.
Kind of like that to try to keep at the right zone.
Gotcha. Thanks.
This idea of the gonadotropin releasing hormone pulsing, nobody knows exactly why, but we do know that it has to pulse. Like if you just have a sustained level of the gonadotropin releasing hormone, it doesn't function properly. So the pulsing is necessary. All right. So that is production of testosterone in the testes. The other thing I want to talk about a little more is the production of sperm in the testes, which is happening in the seminiferous tubules. So for that, I'm going to have to get to another picture because this is getting too messy. So like I said, you've got all these seminiferous tubules in the testes. And actually, you've got, I think I calculated once, you've got something like half a mile of them. They're a lot. So you can make a whole lot of sperm. Like a single ejaculation has hundreds of millions of sperm in it. So we have our little seminiferous tubules. What I'm going to do is, let's do a cross section. So this is cross section of the seminiferous tubule. So like taking one of these things and just cutting it and looking at what does it look like. It's got these cells that make up its wall. And again, this is the lumen. This is the inside. Somewhere out here are going to be interstitial cells. Right? Remember, interstitial cells are the cells outside of the tubules. So the walls are made out of these things. They can be called nurse cells or Sertoli cells or sustentacular cells. But basically, they kind of envelop the little developing spermatocytes. So all those little spermatogonia that I talked about, they're kind of more on the outside. And then those are kind of committing, and then you have the spermatocytes, and finally the spermiogenesis. By the time you get into the lumen, that's where we have the actual little sperm with the tails forming. So this is spermatogenesis, happens kind of going from the outside down into the lumen. And these Sertoli cells are what provide special paracrine factors and stuff that keep the little spermatocytes going along the path. So these Sertoli cells are kind of enveloping and also providing different factors that promote the development of the sperm. So, what hormone is telling the Sertoli cells to keep spermatogenesis happening?
It's not luteinizing hormone?
Now, luteinizing
No,
hormone is the one
it's just for testosterone.
just for testosterone.
The follicle stimulating hormone?
Uh-huh. So FSH is talking to the Sertoli cells and telling them to promote sperm production. If we have adequate sperm production, there's another negative feedback loop. These can send back a hormone called inhibin, which is, again, is going to be negative feedback. So if enough sperm or sperm production is high enough, we have more inhibin, and the inhibin is going to talk to the pituitary and say don't release as much FSH. And it's the same basic idea. It's going to keep the sperm production kind of in that sweet spot. If it's getting too fast, the inhibin will be higher, which will slow things down. If it's too slow, there'll be less inhibin, which means more FSH to speed things up. So it's just another one of these negative feedback loops where the more you've got the sperm production, the more inhibin slows down the signal to promote sperm production. So does that make sense? And I should mention, I mentioned just a few moments ago the idea of the blood-testis barrier. We talked about the blood-brain barrier, how the vessels in the central nervous system have a much tighter control about what comes in and out of the capillaries and all that. You have a similar thing going on in the testes. Again, one of the reasons I mentioned is that the sperm are so highly differentiated from a normal body cell that you kind of want to hide them. So you don't want your antibodies and stuff to see them. Another thing is, in order to have sperm production happening, you have to have very high levels of testosterone in the tubule. So there's this like androgen binding protein that keeps really high levels of testosterone in this region, and you don't want that kind of leaking back, diffusing into the bloodstream. So there's different reasons why it makes sense to have these specialized kind of more impermeable vessels down here. All right. So what we're going to do now is kind of put this in the bigger context of the male physiology and talk about now not just how do we make sperm, but how do we kind of mix them up with all the stuff and get them connected up with the egg. Okay, so again, we have the testes, but now we're going to put in the whole rest of the person. Again, the testes are kind of sitting here in the scrotal sac, and we're going to need a penis. I'm assuming you know there's two testes, but just to make life easier, I'm going to draw one here. This is the abdominal cavity here. Right, so the other thing we need to include in our picture, in addition to the testes and the penis, is we got to have the, the penis has got the urethra, which is coming up into the bladder. Not 3Ds. There. Here's the urinary bladder. I talked about there's the epididymis and then the vas deferens. The vas deferens goes through a little tunnel called the inguinal canal to enter into the urethra. That's how the sperm are ultimately going to make it out the tip of the penis, is this vas deferens burrows through this little inguinal canal. And gets up there. This is a really common site of hernias, an inguinal hernia. Hernia is when something is kind of squishing out someplace it shouldn't be. So if this little tunnel gets stretched out, you can actually have like intestines, your intestines like pooching out into your scrotum. So that would be like an inguinal hernia if you have like intestines from inside your abdominal cavity spushing out through that little tunnel. Obviously, the other things that go through that tunnel are going to be blood vessels, right, to supply the testes. Veins that are bringing the blood back up. There's going to be nerves. All of those things together, the blood vessels, the vas deferens, the nerves, we just call that all collectively the spermatic cord. And there's actually a really cool way that this kind of pre-cooling thing, like the testicular artery is bringing kind of blood down to the testes. And then the blood that's returning back up kind of is in this kind of network of veins that surround, they call it the pampiniform plexus. The basic idea here is the blood that's coming in is coming straight from inside the body, so it's at body temperature. But the blood that's returning, that's been hanging outside the body in the little air-cooled scrotum, is already cooler now. So this vein blood is surrounding around the artery and pre-cooling the arterial blood as it's coming down to arrive at the testes. So this pampiniform plexus kind of pre-cools the blood as it comes down and enters down into there. Other things that, again, help with this temperature control. There is this big muscle called the cremaster muscle. The cremaster muscle attaches to your abdominal wall and basically can pull the whole scrotal sac, pull the testes up close to the body. So it can either keep things warmer by keeping it against the body or cooler by letting things dangle more. This is also triggered by the sympathetic nervous system. Right? If you fight or flight, you're going to want to try to pull the testes out of danger so they get pulled up right up against the body, less likely to get hurt. There's another muscle I just mentioned, the dartos muscle, which changes the amount of wrinkle on the skin so you can have more or less surface area to radiate out heat. So there's also kind of a superficial muscle, the little dartos muscle. So you've got different ways to control the temperature here. So in terms of the actual semen, the stuff that gets ejaculated, only about 10% of the total volume is fluids and sperm that are coming from the testes. In fact, only like 1% of the total volume is sperm. It's mostly other stuff. So what we're going to talk about now is the other 90% of the volume of the ejaculate, of the semen. And it's coming from two different glands. And again, I'm drawing it on just one side, but assume that everything is bilaterally symmetric. So there is the seminal vesicles. Get rid of this stuff here. So these seminal vesicles, they're kind of on the kind of back of the bladder, and they are donating a couple of different things. These are about maybe 60% of the semen is from the seminal vesicles, 60% of the volume. Fructose, right? I talked about how sugar is going to be helpful because these little dudes need energy to swim. Fibrinogen. Why would there be fibrinogen? What does fibrinogen do?
Clotting or help with clotting?
It's that clotting. It makes that kind of... So this is basically after ejaculation, the semen actually kind of gels. And the basic idea is, it wouldn't be so great if a guy, if the ejaculate went up and then just like poured right out. So once it gets ejaculated up into the female reproductive tract, it actually kind of gels to kind of hold in there to give the sperms a foothold to kind of continue on their way. There are prostaglandins. One of the things that prostaglandins do is actually can cause the female reproductive tract to kind of have peristalsis upward, to kind of throw the little sperm up. They don't have to swim the whole way. The female reproductive tract actually helps throw them further upwards. And so that's seminal vesicles. And then the other gland that is making, like I mentioned that, maybe the testes are making like 10% of the semen. 10% of the semen is from the testes. 60% of the semen is from the seminal vesicles. And then the other 30% of the semen is going to be from the prostate gland. The prostate gland is kind of like a walnut with a hole in the middle that surrounds the urethra here and has little ducts that kind of dump into the urethra. Um, it gets its name because people used to think it was the source of the prostaglandins, and now they know that actually the seminal vesicles are the ones that make it, but the name's the name already. The main thing, prostate gland has stuff that capacitates the sperm. And I'll mention what that means in a second. So when sperm are getting made, they're in this kind of state of suspended animation. Right? I was talking about over here, we've got like the sperm are getting created, and here they are in the lumen here. And then from the lumen, they're going to end up in the epididymis. It doesn't make sense to have these things just bouncing off the walls, like ready to go, ready to go, ready to go. It's going to be a waste of energy. So they're kind of just like asleep. And in order to fertilize the egg, they got to wake up. And that's what that capacitation is. It's kind of waking up these sleeping sperm. So, the sperm don't get to the penis by swimming. The sperm get to the penis by peristaltic waves coming up the vas deferens and stuff. It's not till they've been ejaculated and they're mixing with this stuff that they wake up. And it can take hours. It can take 2 to 8 hours to fully be functional and able to fertilize an egg. So that's capacitation, is kind of waking the sperm up from their slumber. So this is the basic idea here. The actual ejaculation is a sympathetic reflex, and the basic idea is we can do like in ejaculation, the first step is what's called the emission phase. I just spelled that wrong, I think. So in the emission phase, this is where the guy knows he's coming. He can't stop it. He can't not come, but nothing is squirting out the tip of the penis yet. But it's all in motion already. And what's happening here is, if you remember, the urethra had an internal sphincter and an external sphincter. Remember, there was the internal sphincter. I'm going to draw it here. This is the internal sphincter. And here's the external sphincter down here. So the skeletal muscle external sphincter, the smooth muscle internal sphincter, they both pinch off, and they isolate this little area called the prostatic urethra. And this is where all of the parts of the semen are now mixing together and kind of swelling out this little area. They call it like the ejaculatory bulb, as it's kind of swelling up. And again, you've got fluid and semen coming from the testes via the vas deferens. You've got all of this stuff, the seminal fluid coming from the seminal vesicles. You have this prostatic secretions coming from the prostate gland. All three of them are all dumping into this zone here. Again, like I said, the guy knows he's coming, but nothing's squirted out the tip yet. And then we have what's called the expulsion phase. And the expulsion phase, that is when this, too much. That's when you relax the external sphincter. And there are these muscles at the base of the penis that actually contract, that squirt everything out, like the bulbospongiosus muscle and stuff like that. So that is called the expulsion phase. So that's the actual, like the semen squirting out of the penis. Um, the other thing I should bring into this story is obviously the penis and the urethra are used for the urinary system and for peeing. So there is actually a gland that actually kind of cleans out the lines during sexual arousal, kind of getting things ready for this whole process. So, kind of at the very base of the urethra here, kind of, I'm trying to draw this nice. I'm going to actually erase some of these other words here. So these little glands I'm drawing right here at the base of the urethra, these are called the Cowper's glands, also called the bulbourethral glands. And they make this mucus, like during arousal. So this is like, pre-cum, right? If when a guy is getting sexually aroused, there's this kind of little slippery, mucusy drop that shows up at the tip of the penis, right? What that is these Cowper's glands basically kind of clearing out the urethra and kind of getting it ready for the eventual kind of semen coming down the line. So that makes sense. The little Cowper's glands. Finally, the last thing we'll mention for the guy is obviously for copulation, the penis has to get hard. So it can get up into the vaginal canal and all that. So let's talk a little bit about that process, about erection. And the basic idea here, like, let's see, penis, urethra. So the head of the penis is called the glans. Glans actually means acorn, especially if if a guy is uncircumcised and there's still the foreskin there. It looks a lot kind of like, you can see why they called it like an acorn, right? It's kind of like the cap on an acorn, an acorn. The glans has a whole lot of nerve endings, for sexual pleasure. And we're going to be talking more in a little bit about the homologies between male and female. Like both, whether or not you're going to ultimately be a little boy or a little girl, up into about a month and a half, you have the exact same stuff inside that eventually either develops into the male version or the female version. The glands of the penis is what is the glans clitoris in the female, which is also the place where there's all these little nerve endings for sexual pleasure. And there's a prepuce of the clitoris as well, a little foreskin over the clitoris. Then there is going to be the spongy tissue that allows the penis to become erect. So let's put that in here. There's spongy tissue that is kind of in here. It's going to be the corpus spongiosum. There's going to be two more ones that go down the kind of shaft of the penis, the corpus cavernosa. If we do like the cross section, there is this corpus spongiosum kind of around the urethra, and then these two shafts that are the corpus cavernosa going along the shaft. These are spongy tissue. It's connective tissue and smooth muscle that has the ability to fill up and engorge with blood and swell up. So basically during erection, you stop the outflow and you increase the inflow of blood into the region, and that's going to swell things up. Things are going to, you'll achieve tumescence, right? Kind of swelling. And again, if this is an example of what we call vasocongestion. When we talk about overall sexual response, both male and female, a major part of sexual response is this idea of vasocongestion, kind of increasing blood flow into the region. During sexual excitement, the clitoris also has its own corpus spongiosum, and it is also swelling and becoming more filled and more sensitive. In the guy, you've got these corpus cavernosa, which are swelling and creating kind of the more erection. And these corpus cavernosa, we call the crura, anchor on the pubis. Basically your pubic bone. So that way the erection is not just kind of a stiff penis that's just kind of dangling there, but it's the penis is getting stiff with relationship to the body as well. And the erection is a parasympathetic spinal reflex. Parasympathetic, is part of the reason guys can have trouble getting hard if they're nervous, right? Because parasympathetic is more the relaxed part of things. Spinal reflex, meaning that it doesn't involve the brain for integration. You can have someone who's a quadriplegic, who is numb from the neck down, and there are touch receptors. If you stroke their penis, they can actually have an erection just from that mechanical stimulation integrated in the spinal cord, responding by causing vasodilation and vasocongestion, even though the guy doesn't feel anything. So what else? I think that's going to be it. Oh my God, we're going to have to, because we still got the female stuff to do here, but luckily we have time. The lab exam is not a 3-hour exam, so don't worry about that. So let me double check if I've missed anything I wanted to talk about the guy. Oh, I should. Okay. What is the typical procedure for male sterilization?
Vasectomy.
The vas deferens.
Yeah, the vasectomy. So in a vasectomy, you basically go in and you cut the vas deferens. This is something that can be done like outpatient, because you can actually access this through the scrotum. You can actually just make a little incision in the scrotum and reach in, pull out a little bit of the vas deferens and tie it off there. What that means. What is the ejaculate going to look like for somebody who has had a vasectomy?
It'll look normal.
Going to look regular, because again, 90% of the semen is actually the stuff from the prostate gland and the seminal vesicles. It's just if you look at it under the microscope, there aren't going to be any little wiggly sperm in there. I should mention, every once in a while, the two ends do find themselves and can repair themselves without you wanting to. That does happen once in a while. Sometimes when people are trying to reverse them, it's possible, although it should never be thought of as a reversible form of birth control, because you never know for sure that it's going to be possible to repair it, although sometimes they are able to. And again, the sperm are still getting produced, but they just basically, if they're hanging out long enough, they just get phagocytized in the epididymis and get recycled. So it's not like, the testes are going to inflate and explode with sperm if they don't have any place to go here. So, I want to mention that. What else? Okay. This is just a silly little thing. I kind of calculated from some of the data, like how fast the semen is getting sent out of the tip of the penis during ejaculation, and it's somewhere around 11.5 miles an hour, something like that. But anyway, let us continue. So I'm gonna kind of expedite things a little bit by sharing some of the PowerPoint images, which I will upload later this afternoon, so you have this on the website, on Canvas. And also, when I was looking at this, I realized there's one extra little thing I should mention about the male reproductive system before we leave it completely. And that has to do with the prostate gland here. So, and this is kind of showing what I was saying, like the vas deferens coming up, the seminal vesicles, and then here's this prostate gland, which is, like I said, it's kind of like a walnut with a hole drilled down, which has the urethra going through and is pouring its secretions into the urethra. What is going to be a problem if somebody has an enlarged prostate gland?
They cannot pee or will have problems to pee.
Yeah, exactly. So it's a very common thing. As guys get older, they call it benign. I don't know if I'm going to draw it well. Benign prostatic hypertrophy. Benign meaning it's not. There is prostate cancer and all that. It's a whole other kettle of fish. But a more typical thing is just the prostate gland gets enlarged, and as it gets enlarged, it pinches off the urethra, makes it trickier to pee. That's something that happens. Oh, yeah, this always cracks me up. He always looks like he's about to say something like, "Hi." So this is just showing the corpus cavernosa, the rods of spongy tissue along the shaft of the penis, and then the spongy tissue that's in the glans penis as well as around the urethra. This is also useful just from a clinical standpoint, showing this is the rectum here. and going up into the colon. Here's the anus, obviously. And here you can see the prostate gland. So this is a very common way to assess for enlarged prostate or health of the prostate. Is actually the digital rectal exam. Like the provider will put on a rubber glove and stick their finger up through the anus, and you can actually palpate the prostate gland right there through the wall of the rectum. So that is that. So now we're going to get back to the female side here.